Hi, this is Miss Nikki. Let's talk about Chapter 5. So basically, let's talk about the plasma membrane structure and function. There's a few key terms that you have to be able to understand. I'm going to skip to the next slide here and then we'll go back. These are all the different types of proteins that we can have that are embedded within a plasma membrane. Let me get the pen working. So plasma membranes have different cells and they have different proteins that are going to carry out specific jobs. So go back to this picture. Let me get everything going here. This does not want to behave. All right, close. We'll put it in picture form. Okay. So these big blobs right here, these are the proteins that I just showed you. So these are proteins. What do we know about this? This is our plasma membrane, right? This is our phospholipid bilayer. Phospholipid bilayer. You might see these terms, hydrophobic and hydrophilic. The heads like water. Philic means like, so they like water. The tails, remember these are fats, these are lipids. They're, I don't know if you can see that. They are hydrophobic. Phobic means fearing, so these guys hate water. So basically, if you take a bunch of phospholipids and throw them together in a fluid, in a solution of water, they will automatically form this phospholipid bilayer. The tails are trying to get as far away from water as possible, and the outside, the hydrophilic heads, like water, so they're trying to get as close to water as possible. So you will come up with this sphere-like structure if you take a bunch of phospholipids and put them in water. Some other things you might hear this called or read it called fluid mosaic model. This is moving, and I do have a video for you to watch in Blackboard. This is not stagnant. This is not rigid. This is not like paper. This is a very fluid, watery. These proteins can drift and move around within this plasma membrane. So it's movable and flexible. Some of the other terms that you should be familiar with are glycoprotein and glycolipid. So glyco means glucose. And protein is a protein. It's made up of amino acids, right? So a glycoprotein is a protein that has a sugar tag attached to it. And so a glycolipid is when you have a lipid, a fatty acid tail, with this glucose attached to it. Okay, so here again are the different proteins. We have channel proteins, we have carrier proteins, cell recognition, receptor, enzymatic, and junctions. So I thought I'd show you a picture. So basically, if we want to move an item from the outside to the inside, we're going to have a channel protein. Usually these are for small non-charged molecules like a CO2 or an oxygen. We might have a carrier protein where this has to actually bind and then be brought into or carried inside the cell. These are called cell recognition proteins. I just showed you here is a glucose, and here is a protein, so this is a glycoprotein, right? This carbohydrate tag, you see how it's 1, 2, 3. Well, somebody else's might have 1, 2, and then have a 3, and then have a 4th over here. So everyone's carbohydrate chains on their glycoproteins are different, and that's what allows your body to differentiate between something that is foreign or something that is natural. So your body can say, oh, that's a bacteria cell, we need to kill it and get rid of it, as opposed to, oh, that's a Miss Nikki cell, we want to keep it. It's doing its job. Receptor proteins. Basically, if you have something that wants to bind and you want to have a signal sent inside the cell, hormones work this way. So the hormone is too large to get through the plasma membrane. It basically docks and then it sends a message or a signal out to the inside of the cell. Enzymatic proteins. We briefly talked about this when I talked about sucrose and sucrase and breaking down products. We're going to talk about it a little bit more. But basically, if I want something to bind and I want this protein to break this product down into a different product. Think about dehydration or hydrolysis. Those were enzymatic reactions. And then this last one is called a junction protein. And junction proteins, we'll see a little bit more in just a second, but basically this is one cell and this is a second cell. If we want two cells to stick together, we have to have a way for them to connect, and that happens through these junction proteins. Again, think about all of your individual heart cells. All of your heart cells have to act together at the same time to beat your heart regularly so you can pump blood through your body. So you have to have cells that are anchored or junctioned or put together in some way, shape, or form so they can all behave in the same manner when needed. Okay, some little more difficult concepts to talk about. This is a little bit more physiology kind of related. We're going to talk about diffusion and osmosis. So these first two require no energy. So diffusion, no ATP needed. Remember, ATP is the energy we use to do work in the body. It's based on what they call a concentration gradient. Basically, items like to move from high to low. So moving from high to low, and this is a molecule, and we call it passive transport. So in this example, they have a membrane here, and I guess I should be clear that diffusion is how we move things in and out of a cell. Remember, I said cells have to take in nutrients and they have to get rid of waste. So we have to have ways to get items inside the cell through the plasma membrane. So this is supposed to mimic a plasma membrane. And they're showing you if I add some red dye to this side, these are all the little dye molecules, how long does it take for the dye to move over here into the water? They're going to even out, right? See this tiny little space, this little hole? It allows for the water molecule to move right through. Or I shouldn't say that, the color, the dye molecule to move right through. So, diffusion. No energy necessary. You're moving from high to low. Do we see how the concentration is high here and it's low here? So all the dye molecules are going to move over to the low side. Another way to think of this, think of a tea bag. If you put tea bags in water and you let it sit outside, what happens? The tea molecules within the bag disperse into the water and you make the tea. Think of it if somebody is smoking in the front of the room. They have tons of smoke molecules right around them. Eventually, what happens to the smoke? If you're in the back of the room, the smoke eventually, you can smell it, right? So the smoke dissipates from the high concentration to the low concentration in the back of the room. The best example of diffusion is when we move oxygen and carbon dioxide in and out of our lungs. So we want to breathe in oxygen and we want to exhale the carbon dioxide, and we do this by diffusion. Thankfully, it doesn't cost us any energy. We don't need to use energy in order to breathe in oxygen and breathe out carbon dioxide, although we do use some muscles, and those muscles do need energy. There's something called facilitated diffusion. If I want to move molecules from a high concentration to a low concentration, they might have to bind a very specific enzyme or protein and then move across the membrane. So you can see that this protein is designed to fit this molecule and this molecule only. Still no energy. Osmosis is talking about moving water across a plasma membrane. Diffusion was moving any molecule that's non-charged, like carbon dioxide or oxygen. In osmosis, if the molecule is too big to move, we will move water to try to balance out the concentrations. Okay, so let's look at this, which is trying to show you osmosis. And they're talking about solutions, so I'm going to draw little beakers. So there's water in here. And osmosis you have water moving from a high concentration to a low concentration, no ATP necessary. So this is called still passive transport. All right, so in this example, isotonic, which means same, they're equal. The water outside is equal to the water inside. In a hypotonic solution here, the water from the outside is going to rush in to the inside of the red blood cell, going from high to low. That's going to cause the red blood cell to swell and burst. In the hypertonic example, the water from the inside of the cell goes outside to balance, and in this case, the cell is going to shrink. Sometimes it's better to look at a particular substance to kind of get a good idea of what's going on here. So let's say we have salt inside and we have salt outside. We're also going to have water inside and we're going to have water outside, right? They're saying those concentrations are equal. In a hypotonic solution, the sodium chloride on the inside is high, and let's say the sodium chloride on the outside is low. Well, remember there's water out here, right? And there's water inside. Now the sodium chloride is too large in order to move in and out of the red blood cell. It can't get in and out through the plasma membrane, but the water can diffuse and equal these two amounts out. So what happens is the water here is going to go inside the red blood cell and try to bring this sodium chloride level down. It wants to make these two, the inside and the outside, equal. In this example, and obviously when the water floods inside the cell, that causes it to swell and sometimes it can burst. In this example, in the hypertonic, the sodium chloride inside is low, the sodium chloride outside is high. So where does the water go? The water is going to go from inside to outside to try to bring that sodium chloride level down. So that's going to cause the water to leave the cell and try to make these inside and outside equal. That will cause the cell to shrink or to shrivel. In animal cells, it's not quite as detrimental as it is in plant cells, it's not quite as detrimental as it is for animal cells because plant cells have that cell wall. So you can see the cell wall doesn't shrink, just the inside shrinks. So again, not as detrimental to a plant cell as it is to an animal cell. Okay, so now this is the opposite. This is active transport. So the other one we did was passive, right? Passive was diffusion, osmosis, no ATP, everything's moving from high to low concentration, right? This is active. Well, active, you're going to be moving from low to high, and you need ATP in order for this to occur. So this example is trying to show you that ATP needs to bind to this enzyme to allow this molecule to be brought in. And when I talk about low concentration versus high concentration, there's 3 molecules over here as opposed to 5 here. So this is low and this is high. You're moving from low to high. The best way to kind of think about this is if you're trying to swim upstream. That's what active transport is. So active transport, you're swimming against the current. You have to put in energy. So you need energy to get upstream from low to high. Passive, you're floating on your back. So no ATP needed, you're going from high to low. There's another type called bulk transport. So if something is really large, we can't squeeze it through the plasma membrane, we can't diffuse water across a membrane through osmosis to equal out the concentrations. It's a really large item, and we either need to get it in the cell or we need to get it out of the cell. So some of the words you should be familiar with, go back and look at endocytosis, exocytosis, phagocytosis, pinocytosis. So endo, bringing something in, cytosis, cell. Exo, out, cell, right? Phago means to eat, so cell eating. This occurs when material is taken in that's really large. Pinocytosis is usually refers to a liquid, if we're going to take in a liquid substance through this process. So here's a great example. The plasma membrane engulfs the item that it needs to bring in, and basically the plasma membrane forms a vesicle around the item, and then this gets brought into the cell. So this is outside, and this is inside. And this does occur where we need to bring items inside the cell that are really, really large. Maybe proteins, maybe a food source. Okay, the last couple things I want to talk about are some of these communication junctions that we briefly talked about. So there's something called a plasmodesmata. This isn't a plant cell. So plasmodesmata is what connects plant cells to each other so that they can communicate. In animal cells, again, make sure you know the difference. In animal cells, we have anchoring junctions, tight junctions, gap junctions, and we have this stuff called extracellular matrix that I'll talk about in just a second. Let's look at the pictures. So a tight junction, it does just what it says it does. It keeps the cells so tight together that nothing can pass or penetrate between the two. So probably the best way to think about this, or where it would be located, would be in the stomach. Do you want digestive hydrochloric acid to leak into your abdominal cavity? No. So you're going to have tight junctions to keep the acid, hydrochloric acid, in your stomach where it needs to be to help digest food. Anchoring junctions are going to help two cells stick together, basically. Probably one of the best ways that you'll see this is in your skin cells. So your skin cells have the flexibility to move, and again, if your muscles are contracting or you're twisting your arm, your skin is moving with those contractions or movements, but the cells are still anchored together. And one of the ways that we can see this is if you ever have a sunburn and you peel off a layer of your skin, it comes off in sheets. It comes off connected because of these anchoring junctions. And then the last one we have, they call them membrane channels. Sometimes they're called gap or communication. So do you see how there's tiny little holes that run all the way through the center? So these communication or gap junctions are very helpful in cells communicating so that they can act at the same time. So that example that I gave you about heart cells, your heart has to beat all in one rhythm in order to pump the blood into your blood vessels. So the heart cells all need to be on the same page and have the same information. So you'll find lots of these communication or gap junctions in heart cells. The last thing they want to mention is for you to realize there's something called extracellular matrix. So this is inside the cell, and this is outside, and sometimes they call this extracellular matrix. So remember how we had our DNA, and then we had our RNA, and then we made our protein, and then I said sometimes those proteins are shipped out of the cell. Well, guess what? All of these are proteins that were made by the cell and shipped out of the cell, and this extracellular matrix also helps two cells stick together. So this makes like a sticky adhesive that allows cells to help stay together. All right, energy. Let's talk about a little bit about energy. These two you must know. What's the difference between potential and kinetic energy? So potential is stored, kinetic is energy in action. If you eat food, you're storing the energy that's in the food molecules. When you run or climb up a hill, you're using that energy in action or kinetic energy. There are two laws of thermodynamics that you should at least be familiar with. Energy cannot be created or destroyed. but it can change from one form to another. But if you change it, you're going to lose some of that energy, and usually as heat. So you can change energy from one form to another, but every time you do, you're losing heat in the process. So we can go back to this picture. We had a similar picture when we looked at producers and consumers and decomposers. I don't remember what chapter, 2 or 3. So here is the sun, and what did we say the plants do? The plants take the sun and they turn it into glucose, right? Well, to take sun energy and turn it into glucose energy, you're going to release heat. So we're taking one form, we're turning it into another form, and we're losing heat in the process. Then what do the animals do? The animals take the glucose and we turn it into ATP, but when we do, we lose heat as well. Now, if we take this chemical energy, let's say this is glucose, and we're going to turn it into ATP, they're telling you you're going to release heat at the same time. So in order for this conversion to occur, you're going to generate heat, and this actually helps generate our body temperature. Helps maintain and generate our 98.6 body temperature. Okay, so cellular work is powered by ATP. We have to look at what ATP is and how it works in a circle or cyclical fashion. They call it the ATP cycle. These are big fancy words, but you're going to see in just a second. Enters, ender, exits, exer. Okay, so this is ATP. Remember, this is the base, adenine. This is the sugar, ribose. And these are the 3 phosphates. And what's stored in these bonds is energy that we stole from glucose, right? All right, so if I have ATP and I break ATP down into ADP plus P, what are they saying? They're saying, what happens if I cut this bond? If I cut this bond, what comes out? Energy. So I take ATP, I break it down into ADP plus P. What has to be released? Energy. That energy can go into another reaction and cause something to happen. Now, how do we make ATP again? If I have ADP and P, and if I put energy in, what do I get out? I get ATP. So there's lots of ways that you can write this. You can also say ADP turns into ADP plus P plus energy. And this equation can also work backwards. I can take energy plus phosphate plus ADP, and I can turn it into ATP. So they call this the ATP cycle. What you need to understand is ATP breaks down into adenosine diphosphate plus this phosphate, and every time that happens, energy is released. If I want to take these two and put them back together to ATP, all I have to do is add energy. Sometimes we're going to couple reactions. So I just wanted to show you this picture. If I have C and D and I want to turn it into a different product, we can use ATP, we can add energy, and we can make this reaction occur. And this is leading up to where we're going to talk about photosynthesis and cellular respiration. This is one way in which we can use ATP energy. So ATP causes this myosin head to contract, and that pulls on this actin, which is here, and this is myosin. And the muscle was long, and now the muscle is short. We have gone from a relaxed muscle to a contracted muscle. And what did the contracted do? The contracted allowed us to do work. Lifting this 10 pounds we have to generate enough force to lift that weight up. That's doing work within the body. And what did we need to do that work within the body? We needed ATP. So we've talked a lot about proteins. I want to make sure that we know what an enzyme is. So an enzyme is a protein with a job. Basically, that's it. It has a very specific job. Enzymes can lower the activation energy and make a reaction happen. So what they're trying to show you is if the enzyme is not present, you have to put this much energy in. If the enzyme is present, you only have to put this much energy in. So this is a harder reaction to have occur. If you have an enzyme, all it does is make things easier, right? Here is an enzyme. Remember, enzymes are also proteins. So this was made from RNA and this was made from DNA, right? So the cell decided it needed to make this enzyme. I have this polypeptide here. It's going to bind to the enzyme, and you see how it's causing it to bend? Well, by causing it to bend, it allows this bond to be broken more easily. That's what they mean by lower the energy necessary, and sometimes they call it the activation energy. Now we have products, and our enzyme can actually go and be used again. So in this one, we went from a 4 molecule to two molecules, right? Molecules. Where have we seen something similar to this? Do you remember dehydration and hydrolysis? Either putting molecules together or pulling molecules apart. There are some factors that can affect how well enzymes work. So basically, substrate concentration. Enzyme activity increases as the substrate concentration increases. Substrate is what went in. Let me just go back one. So this is the same thing. This thing right here, as it binds, it's going to bind right here. That's the substrate. So what they're saying is the more of these you have, the more of this you get. The more 4 molecules you put in, the enzyme's going to work, and you're going to have more 2 molecules come out. Temperature. As temperature rises, enzymes work more quickly. But remember, if you change the temperature too much, the enzyme stops working altogether. Probably the best example of this is a fried egg. When you have an egg, it is a possible chicken embryo, right? It's an egg that could have become a chicken. And the white stuff was there to help feed the developing chicken should the egg have been fertilized by sperm. When you fry that egg, it goes from a snotty white-like substance to this hard, white, opaque substance. You've changed that protein. It was food for the chicken. Now it's this hard protein. It can never go back to being chicken embryo food. You've altered it by temperature to the point that that protein can never do its job again. pH can also affect enzymes. The reason I mention this, stomach pH is about 2. So the enzymes in the stomach like the environment of a pH 2. If you took enzymes from the stomach and you put them out someplace else in the body, the enzymes are not going to work as well. They're designed to work in a pH 2 environment or a very acidic environment. The last thing that affects are cofactors. So we're saying how well an enzyme works. Basically, this is what we're saying, how well the enzyme works. There's something called a cofactor or a coenzyme. And we'll talk a little bit more about cofactors when we move through photosynthesis and cellular respiration. But basically, these are needed to help the enzyme do its job. So basically, let's say we have an enzyme and it needs a vitamin to bind right here, and then this enzyme can increase its job or function, and it can work harder. But without the enzyme, it either won't work at all or it won't work as well. This is why vitamins are important in your diet, making sure that you eat varied amounts of food, because many vitamins serve as cofactors or coenzymes in order to make your metabolism function better or more accurately or more quickly. This last slide is trying to show you how sometimes we have these cascades, is what they call them. So you have one enzyme, enzyme A, all right, let me rephrase that. A binds enzyme 1, and the product is B. Now another enzyme binds B and turns it into C, and then a third enzyme turns it into D, then a fourth turns it into E, and then we have our final product at the end. So this will keep happening until you have enough of your end product. Then your end product will go back, it will bind this enzyme, change the shape, and now A cannot bind that enzyme. So we have some really complicated metabolic pathways. If we need to make a whole bunch of F, we need to make 100 of F, but we don't want to make any more than 100, we will continue this production line until we reach our cap, and then we'll go back and we'll shut off this assembly line. Kind of similar to how a heater works, right? If your house is cold, you turn the heater on and the heater churns out heat until the inside of your house reaches a certain level, and then it goes back and shuts the heat off. So we do this in our body all the time to regulate some of these complicated metabolic pathways that we have. All right, on to photosynthesis.